Hello everyone, so here we are with our rat dissection. Becca and Raylin are going to be partners for this rat dissection. Due to COVID, of course, you need to be wearing protective equipment. For any dissection, they've got their gloves and apron. Additionally, they've got a face shield, since they will be working close together, as well as face masks. The rat itself will be given to you either from a pail or a plastic bag as here, and then each of the students has selected their different roles. Becca will be doing the cutting. So Becca, talk me through your equipment here.
Here I have my razor, which I'm going to be using to cut, along with the scissors. I have my dissecting pin for moving things around, the organs once I get inside, and my tweezers for moving as well.
And Raylin, how about you?
I also have a set of tweezers in case I need to move the skin flaps, because that is my whole thing, is I'm going to be pinning back the skin flaps. So of course I have my pins and my scalpel in case the skin gets stuck and I need to cut it some more.
Perfect. Now, of course, you all should be familiar with the orders of the cut from the dissection preparatory manual. And hopefully you've spent some time with our rat model. Now this rat model is a cartoon of what we see. So now it's time to take a look at the internal structures of the rat. Two other important notes about the dissection today. These were living animals. Treat them with respect. I do not want to see any horseplay. Treat them with dignity, because they have passed away for your educational purposes. Second, any rat wastes need to be disposed of in the singular trash can that will be in the center of the room usually, so somewhere central to the classroom, that is double bagged. This is for the courtesy of the folks who will be cleaning up after us. Do not put waste in any other receptacle. So Becca, let's go ahead and open up that bag and see if we can make our first incisions.
Out of the way. Maybe.
I think you're gonna have to cut into where the air sac is, yeah.
That's what I'm thinking. This is much easier with the ones from the pail.
Yep. So we'll get the rat opened up and then see you back here to record the first cuts. All right, we've got the rat out of the bag. Now Becca's ready to make our first incisions. Walk us through it, Becca.
All right, so first I'm working to, usually you could separate the skin and the muscle, and that makes it a little bit easier if you massage it a touch. I don't believe we need to in this case. So, first incision is going to be directly from the chest down to the reproductive systems here. And sometimes you have to give it a little bit more love than you would like to, because they've got tougher skin than you might think. However, you don't want to cut too far through, because we don't want to be cutting open all of our intestines.
Yeah. Try to keep the internal organs intact. So first cuts made via the razor. Once tissue is open, cleans up the way for scissors to come through.
There we go. All right, there's our first cut.
All right, but that's not good enough yet to pin open the skin flaps for Raylin to get on that job. So let's make a few more cuts.
Second, we're gonna go across here down by the legs.
Relieve the tension on the skin flaps.
There's one. I have to turn our rat a little bit here so I can get to the other side.
Sure. Yeah, you can move around.
There we go. All right.
Cool. Let's
Second
take a
cut.
quick close look at the space between the skin flaps and the muscle. So Becca, do you want to try separating the skin from the muscle? You might need to use that dissecting pin.
Yeah. We're very correct there.
So you should be able to separate those two different tissues, as there is connective tissue in between them. But that should show you the stark difference between the epithelium and the muscle. Very cool. So now let's get those skin flaps a little bit more open so that way Raylan can pin them down.
All right, the third cut is up between the front legs.
Nicely done.
Get that as open as possible there.
And Becca's stopping before she reaches the throat cavity, so that's perfect.
Right up between the legs. I'll show it again. A little bit more connective tissue, the diaphragm there that's holding that together. Make sure that's open.
All right. So with all those cuts made, we'll see you guys back here once we've got the rat drained of some of the liquids and the skin flaps pinned down. All right, so here we have the rat cut open and pinned. All of the external internal organs, or the top layer of the internal organs, are visible. So using your assigned organ bank, Becca, would you mind pointing out the bladder first?
Our bladder is going to be right down here. Let's move some of the organs out of the way for you. And you can see, it tells you that there's 3 lobes. You can see we've got one part.
So those are the 2 prostate glands. And then adjacent to it, you've got the bladder itself.
Right here.
Very tricky to see, but hopefully it's still there. Oftentimes in the cutting process, down by the legs, some folks will snip it off. So try to find it if you can. Next up, let's identify our large and our small intestines. How do we do that, Becca?
So the small intestine up here.
Thanks, Raylan.
Thank you. So we have both of our sets of intestines. The one, you can see, connects right off the stomach here. Thank you, Paul. Right off there.
And then the large intestine?
The large intestine is all of the rest of this mess.
And then where does it lead to the rectum itself?
Pull that up. You can kind of see down here, it connects right in. Right here.
Yep, so that is the final evacuation before released as excrement. And you actually have an organ poking out right here. We'll talk about this in a little bit. But in order to get there, we have to finish going over the top layer of stuff. So we can put that back down. So all of our intestines, small intestine identified by proximity to stomach, large intestine further down, small intestine absorbing nutrients and doing the last of the digesting, large intestine reabsorbing water. Next up, where's our heart?
Our heart is right up in our chest here.
Very cool. And then Raylin, what are those 2 flappy things adjacent to the heart?
These are the lungs. As we can see, this is our right, and then we have our left, which is kind of hidden behind the heart.
Cool. And now the lungs are pulled down via something called the diaphragm. Where's that at, Becca?
Right here. You heard me earlier when I had to cut it so I could get through here to open up the chest cavity. It's just this very thin piece right here.
And it is that tissue that's going to be pulling down on your lungs to expand, contract, expand, contract, changing the size of your lungs to allow you to breathe via negative pressure breathing. Hopefully you remember that from the lectures. Our last organ is our biggest organ. Raylan, where's our liver?
Right here, this big chunk is the liver.
Multi-lobed organ, very, very cool looking. Keep in mind the liver descends from the same stem cells as epithelium, meaning that it is one of the only internal organs that's able to regenerate itself if damaged. All right, so next up, we're going to remove some of these organs to clear the way so that way we can see what's underneath. So with the organ removal, one of the first things you want to do is get rid of the liver as it's taking up a lot of space. So Raylan is lifting while Becca finds the connective points and is cutting off the different lobes.
How else are we connected here?
Might take a couple tries. You might have to cut it off in a couple pieces, but it will be removed. You can go ahead and set it down outside of your rat to keep track of what it looks like for our final image of the lab. We'll continue by removing some of the intestines next, and then see you back here after we're on to our next layer. All right, so we've pinned back the intestines a bit, so that way we can see the internal structures here, where we'll be able to focus on everything underneath the liver, which begins with our stomach. So where's the stomach, Becca?
Right here.
Looks pretty big, and of course, after the stomach, you have the 3 different components of the small intestines. So hopefully you can go back through your notes and remind yourself of those. And then of course, we do fully reveal the rectum of the large intestine, right there. And adjacent to it, we finally have a clear view on that organ underneath the stomach. That is the spleen. Now, if you lift up the spleen, which is quite large, you should be able to see your first kidney. There's one kidney on this side, and there will be another kidney on the second side. Hold on while we grab a light. So you should be able to see that in there. Excellent. Thank you. Thank you. Now, the only other organ adjacent to the stomach that we have not gone over yet is the pancreas. This is a very thin film. Becca, would you mind pointing out the pancreas to us?
The pancreas is all over right here. You can see that film that Paul was talking about. It's kind of going away.
Kind of dissipating, but you've got the pancreas there. Excellent. Okay, so now we're going to, next up, remove those organs. So that way we hopefully get a good view on the kidneys behind. All right, so now we've removed the stomach, the small and large intestine, the spleen, the pancreas. All of that is sitting adjacent to our rat now. And you can see the kidneys, the 2 kidneys placed adjacent to the spine of the rat. Now, Becca, can you walk us through real quick approximately where you made your cuts to be able to remove the entire digestive cavity like that?
So when I was removing this, you can see it's kind of inverted right now because I have the stomach down at the bottom of the rat rather than up at the top where it would normally be. But I went first underneath the organs, so right in this area, and I pulled them back like we had before when we were observing the spleen and the rectum, and pulled them back and went right along the backbone here and cutting the rectum down here, you can see.
Yep.
And then I pulled it from the opposite way. So whatever was left of the stomach, any of the kidney that was left up there, I pulled that back this way and cut right through here. You can see there was some more. And then just making sure to get rid of the connective tissue as I was going to make sure it was clean.
Yep, so you need to have snipped the esophagus leading into the stomach, which you see descends down from the diaphragm because you need some sort of passageway between there. And the diaphragm obviously builds that wall of the thoracic cavity. Hopefully a better view on that tissue. And now we'll work our way up with the major component of animals done, the digestive tract, because remember we are primarily giant feeding tubes. We now can work to our circulatory, respiratory, and then see about the trachea and some lymph nodes in the neck. See all in a bit after we've cleaned up some of that tissue. All right, so we very carefully cleaned up the space with a few more cuts above the rat's neck because we want to now reveal the trachea, the thymus, and the lymph nodes. So the trachea is the major breathing tube of the rat. You can see it right there as Becca's pointing. Now, you also can identify it from the cartilaginous discs that are present. I don't know if you can hear that. I'll try to get closer. But if you are touching that, it should have a bit of a, like, distinct sound to it because there are discs or cartilaginous rings on one side of the trachea that are not there on the other side. Additionally, you also should be able to identify the thymus, the organ directly above the heart that stores very particular white blood cells, the T cells. It's the barracks, as we'll talk about in that lecture. Now, the last thing that's kind of tricky to identify are the different lymph nodes. Here you want to use the stain to help you out, because the lymph nodes are oftentimes adjacent to blood vessels, which here are stained in blue, and they are oftentimes kind of whitish sacs that are collecting water from those blood vessels. So you see a lymph node there. Very easy to cut away or remove. So again, anytime you're operating here in the neck region of the rat, be very, very slow with your cuts, so that way you don't remove things. Next up, we're going to remove the heart and the lungs from the cavity, so that way we can point out the different chambers of the heart. See y'all in a few. All right, so here now we've removed the heart and lungs. Becca, where did you make the cuts to be able to remove them so easily?
On this one again, cuts the top and the bottom. I had to cut it off of the diaphragm. You can see it's now just laying here. And then I had to come up and cut where the trachea connected into the lungs.
Perfect. And you still see the remnants of what I believe to be the esophagus leading down to where the stomach would have been. Cool. So let's take a look at these organs themselves. You can separate the heart from the lungs.
Maybe we will separate the heart from the lungs.
Worst case, you can kind of pinch around it / use the pin. A lot of connective tissue there. Large blood vessels. Yeah, that sounds good. All right, so the heart has been separated. Now, once the heart is separated from the lungs, you should be able to feel with your hands the 4 different chambers of the heart. The 2 atria on top, and then the 2 ventricles on bottom. So this is not something that'll lend itself well to the video, but Becca, if you can just tell us a story of how you're feeling, if you can feel the 2 different squishy chambers on the top of the heart.
They're, like you said, Paul, they're a little bit squishier. They're not quite as solid.
And then how about the ventricles on the bottom of the heart? These will be very tough muscles since they're the pumps.
Nothing moving around down there.
And let's go ahead and see if we can cut the heart close to directly in half to see if we have any view of the chambers themselves. So based on where you're feeling, try to find, and this is very tough, but try to find that central septum that separates the sides of the heart.
It's right in the middle here.
And if you pull that apart, we might be able to see some pockets.
Maybe.
How's that looking there? Yeah, make the full cut. Sorry, Becca.
Um,
A little pushy with my camera work. Okay, and do you see something that looks like a pocket where the atrium might be? Look like we hit the septum? I think so. I definitely see a ventricle down there at the bottom. Nice open space. So hopefully through the aesthetics of actually feeling the heart, you can identify that the heart is a 4-chambered organ. Ask any last questions that you may have during the dissection. But after this, we can remove the pins. The pins will end up in the trash. We, of course, hopefully should photo document our different organs. The liver with its multi-lobed self. One of the lobes of the liver still attached to the stomach, but the stomach and the small filmy pancreas on its bottom. The spleen attached to that. So we see the pancreas, we see the spleen, we see the small intestine, we see the large intestine further down. We see the kidneys within the organ of the rat. Within the body cavity of the rat, the tube that would lead to the rectum, the very clearly now shiny urinary bladder. It does have a bit of a sheen to it, so you should be able to identify it by that sheen. The once upon a time connected diaphragm that was connected to the 2 lungs of the rat that surrounded the heart, the thymus placed above them, and then of course the trachea with its cartilaginous discs on one side, and lymph node collecting the water from the blood vessels. I do hope that this rat dissection was educational, as it's meant to show you some of the comparative anatomy between us and our rodent cousins. All right, so we've now placed the rat and any of its organs remaining in the double-bagged trash bin. Becca and Raylyn are cleaning up the trays with soap and water, as well as any of the utensils that were used. You're going to be leaving the utensils to dry in the central wash bin after everyone is done washing everything. Thank you all for treating the rats with respect during this dissection, and I do hope that they were educational. And thank you, Becca and Raylyn, for being willing to do this.